The TCA, also known as the tricarboxylic acid cycle or the Krebs cycle or the citric acid cycle, lots of names and lots of uses. In its most fundamental sense, we can think of it as a cycle in which we're taking a 4-carbon compound called oxaloacetate, combining it with a 2-carbon compound, the acetyl group of Acetyl-CoA, to get a 6-carbon compound, citrate, which has 3 carboxylic acid groups, so 1, 2, 3, hence tricarboxylic acid cycle. And we can go take this around the cycle, carry out oxidation reactions, eliminate 2 CO2, so we're losing 2 carbons, which gets us back to a 4-carbon compound, that same oxaloacetate that we can take through the cycle again. Well, not the exact same atoms, but the same arrangement of atoms, but we've lost 2 of the carbons along the way. And so if you were to go and track the carbons, you would see that the carbons that you have here are going to be different than the carbons that you have here. But overall, we have this oxaloacetate regenerated, which means that we can keep this cycle going and going. If we keep the cycle going this way, what we're going to be doing, we're going to be generating 1, 2, 3 NADH, 1 FADH₂, which is, remember, this succinate dehydrogenase is your complex 2 of your electron transport chain. And then we're going to be generating a GTP. In some species and some isoforms, this is going to be an ATP, but GTP and ATP are readily interconvertible. So you can think about generating an ATP or GTP, which is basically saying generating an ATP. So these NADH and the FADH₂ can go through the electron transport chain, and you can generate a lot more ATP that way. This is happening in your mitochondrial matrix with that complex 2, remember, in the inner mitochondrial membrane. This is going to allow you to generate a lot of ATP if you use the citric acid cycle in this catabolic manner. We'll also talk about, however, how we can use the citric acid cycle for anabolic purposes. That is for the purposes of building molecules. We can do this if we siphon pieces away from the citric acid cycle for other uses, such as gluconeogenesis, making glucose, or lipogenesis, making lipids. We can also make things like ketone bodies, or we can make amino acids. Lots of cool things that we can do with parts from the citric acid cycle, and we can get those parts into the citric acid cycle from a variety of different sources. So we'll continue on our story of talking about glucose catabolism by talking about how we can get pyruvate converted to acetyl-CoA and take it through this pathway. But we'll also see how we can take components from lipids, we can take components from amino acids, we can take all these different components and enter them into the tricarboxylic acid cycle, and we can enter at various points. It's important that we're able to regenerate these intermediates of the cycle. So normally when we go around the cycle, we're going to lose 2 carbons. We can get those carbons back by bringing in the acetyl group of acetyl-CoA. But if we take something out along the way elsewhere, we're going to have to add more carbons into the pathway. And this is going to get us to think critically about the usage of various components. One of the really great things about the citric acid cycle is that we have these small parts. Kind of like small Lego pieces, you can break down lots of different Lego compounds to get those same small pieces, and you can build lots of different Lego compounds from the same small pieces. Similarly, we can break down molecules into these smaller pieces that then we are able to interconvert more easily. So by having these smaller compounds, these 6, 5, 4-carbon compounds, we're able to interconvert different molecules more easily. We're able to take skeletons from amino acids and bring them in. We're able to take breakdown products of lipids and bring them in. We're able to take these things out and make molecules. So just like those little Lego pieces that we can get from breaking down a lot of Lego complexes, and we can build a lot of different complexes from those little Lego pieces, but it would be harder to build a variety of Lego compounds if you had more specialized kits or you had things already partly built. Similarly, if we have molecules that are big, it's going to be harder to make more things from them. It might be easier to make some things from them because you already have it kind of partly made. But if you want more variety, you're going to be out of luck. The TCA is going to give us a lot of luck on that front because we're going to have a lot of small pieces that we can readily interconvert. So this is where we're going, a more integrated view of the citric acid cycle and how it can be fed into and taken out of for various purposes. But let's start by simplifying things down. Let's continue on our story with that pyruvate that we got from glycolysis. So remember glycolysis, the breakdown of glucose to give us pyruvate. In this process, we are going to be generating on net 2 ATP, and we're going to be generating 2 NADH. If we have the electron transport chain running and we have oxygen, we're going to go through that and we're going to generate ATP that way. We're still, however, going to be left with that pyruvate. And that pyruvate, well, if we look at it, we see that we still have oxidation potential. So remember that oxidation is the loss of electrons. We can pass those electrons onto carriers like NAD plus to give us NADH, which we can use to cash in for ATP by using the energy from those favorable electron hands-offs to power the pumping of protons to power the synthesis of ATP. So oxidative reactions are often going to correspond to catabolism, to the breakdown of molecules and the generation of energy. If we look at this pyruvate, we can see that this carbon is still saturated. It still has 3 hydrogens. This carbon, it only has 2 bonds to oxygen, so it can still bond to oxygen. This carbon, it can still make another bond to oxygen. Remember, the most oxidized you can get is going to be the CO2, such as we see with our citric acid cycle when we're losing things as CO2. That's going to be carbon in its most oxidized form. We're going to have the potential to oxidize that pyruvate more. Doing those favorable oxidation reactions is going to generate energy. So if we want the maximum amount of energy, not only do we need to take that NADH through the electron transport chain and oxidative phosphorylation, but we also need to take the pyruvate further in catabolism. If we look at the structure of pyruvate, we see that it is 3 carbons. And if we look at the structure of the intermediates in our citric acid cycle, we can see that, well, we are never going to have a 3-carbon compound. We go from having 2 carbons and 4 carbons, so we have 6. And then we lose one, so we have 5. And then we lose another one, so we have 4. But we're never going to see something with 3. And so we actually have to lose one of the carbons before we can even get those carbons that came from your glucose into the citric acid cycle. The way that we're going to do this is we're going to be doing oxidative decarboxylation. This is going to allow us to get rid of a CO2 from that pyruvate, so we get that 2-carbon compound, that acetyl group from Acetyl-CoA we can add on. And we're going to be getting an energy IOU in the form of an NADH because we are doing an oxidation. Remember, this carbon, it can form 4 bonds to oxygen, but currently it's only forming 3 bonds to oxygen. So we want to make another bond to oxygen, make this CO2. This is going to involve losing electrons, so oxidation is loss of electrons. Those electrons got to go somewhere. Those electrons are going to go onto NAD plus to give you NADH, which then you can go cash in for ATP. And you're going to be losing that CO2 because remember, carbon can only form 4 bonds. And so if you're forming another bond to an oxygen, you've got to be breaking off this bond. So we're going to lose this carbon dioxide. And what we're going to be doing as well is putting this acetyl group onto CoA. And so this acetyl group from the acetyl-CoA is going to get passed on to our oxaloacetate, which will give us citrate, and then that can go through the cycle. So remember, we have to take that 2-carbon compound and a 4-carbon compound and join them together. It's often difficult to make those carbon-carbon bonds. And by first passing the acetyl group onto this CoA and then passing it onto this deoxaloacetate, it makes things easier. So let's talk a little bit more about what this CoA is. So right now we're just seeing CoA and it just looks like a few letters. But CoA is actually this kind of big handle. And it's got these phosphate groups, which at our physiological pH, these would be deprotonated. So you'll have this negatively charged big molecule, not going to be able to diffuse freely through the membrane, and we're not going to have transporters for it. So if we have a CoA, we're not going to be able to take it in or out of the mitochondria. And so we're going to have separate pools of CoA in the cytoplasm and in the mitochondria. So we're going to have to do this oxidative decarboxylation of our pyruvate inside of the mitochondria. So we take that pyruvate from glycolysis into the mitochondria, and then we're going to do this step. Another note about the CoA is that it is derived from vitamin B5 or pantothenate. So get your vitamins. Okay, now back to the chemistry side of things. Let's think about what's actually the cool part about CoA, because it's not this big bulky thing. It's just little part over here, this sulfur. This is the working end of the molecule that's going to allow a linkage point that allows this CoA to serve as a handle. And I call it a handle because at the end, it's got this sulfur linkage. So sometimes you'll actually see this abbreviated CoAS or CoASH. That's emphasizing that it has this thiol on the end. So remember, a thiol is the sulfur version of an alcohol. And when we think about a sulfur compared to an oxygen, sulfur is going to be right below it on the periodic table. So it's going to have the same number of valence electrons. It's going to have a lot of similar properties, but sulfur is going to have more electrons. It's going to have those electrons further away from the nucleus. So it's going to be bigger and bulkier, and the bonds that it forms are going to be weaker. This allows bonds to sulfur to be kind of swappable. So we can think about having this CoA, having this thiol group be kind of a swappable linkage point. And note here that it says a thioester linkage. A thioester is the sulfur analog or equivalent of a ester. So remember, an ester would be where we had an oxygen next to a carbonyl. And in the case of a thioester, we have a sulfur next to a carbonyl. In addition to forming these weaker bonds, sulfur is going to be very nucleophilic. And so it's going to be able to attack a carbonyl carbon to add on an acyl group. An acyl group is when we have a carbonyl next to an R group. And so in the case of our Acetyl-CoA, we have the simplest R group. We just have this methyl group here. And so this is called an acetyl group, this carbonyl carbon next to this methyl group. And if we had a different R, we would still have an acyl group, but we wouldn't have an acetyl group. And we've seen acyl groups when we talked about our triacylglycerides. So our good old tags are a storage form of our fatty acids. We're attaching those fatty acids through those ester linkages to the glycerol. And those were examples of acyls. And so the acyl there, well, there was the carbonyl carbon, and it was next to this big old R chain, this long hydrophobic tail. Here we have the simplest R group we could have, just this methyl group. And this is an example of an acyl group. And I mention this here not just because I care about you knowing chemical terms, but because you will see this term used. So we've seen it once with our tags, and it also comes up in the case of lipid metabolism when you talk about acyl carrier protein, which basically it is like very, very similar to having that CoA, but it's attached to a protein, and it's going to allow you to have those swappable linkages so that you can pass the long chains of your fatty acids that you're making from place to place, taking advantage of the swappable nature of bonds to sulfur. We're going to be converting pyruvate to acetyl-CoA in the mitochondrial matrix, and we'll be doing it using this enzymatic complex called the pyruvate dehydrogenase complex. Sometimes when we do an oxidative decarboxylation, it's going to be simple, and sometimes it's going to be complicated. This is one of those times when it's going to be complicated. And so when we have these times when it's complicated, we often use these big dehydrogenase complexes that use a variety of cofactors. So remember, those are molecules that the enzyme kind of needs to help it do what it does. So in this case, not only do we need that CoA, and not only do we need that NAD+, but we also need these cofactors TPP, lipoate, and FAD. TPP, this is thiamine pyrophosphate. It's often going to be used when you have these decarboxylation reactions. Lipoate, this is a group that is like this long group with a sulfur attached on the end that's going to be actually held in this enzyme complex. And it has the sulfur linkages that are able to therefore bind to that acetyl group as well. So you can form those thioester linkages to these groups as well. So you're actually going to pass this acetyl group from place to place throughout this complex. And in the process, you're going to pass those electrons onto FAD, which is then going to pass it on to NAD. So it's this big complicated process. And you might be wondering, why do you have to go through all that gymnastics? You have to go through this gymnastics because what we have here is this what we call an alpha-keto acid. And so we have a ketone that is alpha to a carboxylic acid. If we have a beta carboxylic acid, that's going to be a lot easier to decarboxylate. And so when we see beta carboxylic acids, in our citric acid cycle. We're going to see those are going to be easier to break off. But in this case, we have the ketone alpha-2, the carboxylic acid. They're right next to each other. This is going to make it harder to decarboxylate this. And so when you see something like this is going to be one of those instances in which you're going to have to use these more complicated complexes. So for example, if you want to break down an amino acid, one of the branched chains amino acids, that's actually going to have to use a similar complex that's actually going to use some of the same pieces. Because when you deaminate these amino acids, you'll be left with an alpha-keto acid, and then you'll have to go through a similar process to get rid of the carboxylic acid. And here you can see that we have this E1, E2, and E3, which is actually very similar to what we had in the case of our pyruvate dehydrogenase complex, where we had our E1, our E2, and our E3. E1 is the only one that's actually, this is the one that actually binds to the substrate. E2 and E3, these are kind of just where they're passing things around and doing the oxidation reduction reactions. And it turns out that E2 and E3 can actually be shared between different complexes, whereas the E1 will be specific for your substrate. E2 and E3 can actually be used for the same sorts of things. And so we see the same machinery kind of reused in different forms. Let's get back to our story of taking that acetyl-CoA through the citric acid cycle. We saw how we could generate that acetyl-CoA from the oxidative decarboxylation of pyruvate, which we could get from glycolysis, but we could also get pyruvate from other sources, and we could get acetyl-CoA from other sources. So we can get pyruvate from things like proteins. We also know how we could get it from lactate, from fermentation. We could get acetyl-CoA from lipid breakdown. So lots of ways in which we can get acetyl-CoA, and also there's going to be ways in which we can get other components of the TCA. But let's start with thinking of this as a cycle, starting with acetyl-CoA, joining with oxaloacetate. Let's go one time through the cycle, just really quick, to see what kind of like logically what's going on. And then we'll go in more detail to look at the individual steps. We can think about the TCA in kind of two halves. So in the first half, we're going to be losing 2 CO2s. So we're losing 2 carbons. And then in the second half, we're going to be rearranging things so that we get back to our oxaloacetate so we can do this whole thing again. So walk through real quick. We're going to take that acetyl group from our acetyl-CoA, join it to oxaloacetate to give us citrate. That's happening through this enzyme, citrate synthase. We added 2 carbons from the acetyl-CoA. We have 4 carbons from the oxaloacetate. We end up with a 6-carbon compound. And we have a tricarboxylic acid because we have 3 carboxylic acid groups. We're going to lose 2 of them each turn of the cycle. Then in the aconitase step, what we're going to be doing is we're going to be changing the position of this hydroxyl group. This is going to be an isomerization reaction because we're just shifting the position of something. So we can see here we have this oxygen in this position, this hydroxyl in this position, and here we have it in this position. Why do we do this? Well, this is going to set us up for a favorable decarboxylation. We'll get much more into the details, but basically, if you were to oxidize this and get a ketone here, this carboxylic acid would be alpha to the ketone. And as we said, decarboxylation of an alpha-keto acid isn't favorable. We can go through those complicated gymnastics with having a big old complex like we'll have to do later with our alpha-ketoglutarate. But if we can avoid that, let's do it. And so what we can do is we can just shift the position of this OH, and then we're going to, then we'll oxidize it and we'll get a beta-keto acid, which is going to be favorable to decarboxylase. So this iconitase step is going to be setting the scene by moving the position of the OH group so that the isocitrate dehydrogenase can take over. We'll then oxidize this alcohol to give you a ketone. That's going to be alpha beta to this carboxylic acid. It's going to make this favorable to get rid of. Then what's going to happen is you're going to get rid of it. And when you get rid of it, then you're going to get your alpha-ketoglutarate. This, however, is going to be an alpha-keto acid, and we can't just shift things around. So we're going to have to go, and we're going to have to use one of those big complexes. In this case, we're going to use this alpha-ketoglutarate dehydrogenase complex, very similar to your pyruvate dehydrogenase complex. We're going to attach the CoA to where there was a carboxylic acid. Now this is going to leave us with succinyl-CoA. Because we've lost those two CO2s, this part of the molecule at least is 4 carbons, but it's still bound to this handle, this CoA handle. We're going to use this sort of stored energy to generate GTP or ATP and kick off the CoA, leaving us now with just succinate. That was happening with the help of succinyl-CoA synthetase. Remember that although it says succinyl-CoA synthetase, so it kind of sounds like you should be making succinate, these reactions are going to be going in both directions, and so you could make succinyl-CoA from succinate. But in this direction, we're making succinate from succinyl-CoA. And now we have the succinate, we have that step of our complex 2, our succinate dehydrogenase. We're going to pass the electrons from succinate onto FAD. And what we're going to be doing here is you don't see any oxygens, but you do see that you formed this double bond. You've lost 2 bonds to hydrogen, and you've gained these bonds to the carbon, which is more electronegative than hydrogen. So this is indeed an oxidation reaction. The electrons from it are going to the FAD. Remember how we talked about FAD has a higher reduction potential than NAD+, and so we could use it to do this kind of more difficult oxidation. Now what we're going to do is we're just going to hydrate this, so we're going to add water. This step is not going to be an oxidation because yes, we're gaining up onto an oxygen, but we're also gaining onto a hydrogen, so they kind of cancel out. That's not going to be an oxidation overall. But we are going to have an oxidation at the next step. So that step was by fumarase, that was adding the water. Now our good old friend, malate dehydrogenase is going to oxidize malate to give us oxaloacetate. And if we're oxidizing malate, we've got to be reducing something. What we're going to be reducing is NAD+. We're going to take those electrons from malate and form a double bond to this oxygen, generate NADH. So with each turn of the cycle, we generated 3 NADH, 1 FADH2, and 1 GTP. And if we think about going from glucose, we would have 2 turns of the cycle because we would have 2 pyruvate at the end of glycolysis because we do that whole second half twice. So we're losing these carbon dioxides, then we're rearranging things in order to get back to that oxaloacetate so we can do this cycle again just by bringing in another acetyl group. Now that we know the basics, I want to go into a little more detail about a couple of the steps. So if we look at oxaloacetate joining to acetyl-CoA, remember that's going to be with our citrate synthase. If we look at that reaction, it's going to have a really large negative delta G. So here it's showing the delta G not prime, but it's also going to have a large negative delta G. So this is going to be one of the steps where we're going to really want to highly control. Another thing to note about this step is that the enzyme can kind of hold oxaloacetate and plop acetyl group on top of it in such a way that it's going to be able to tell apart which carbons came from the oxaloacetate and which came from the acetyl-CoA. And you're not going to lose the carbons from the acetyl-CoA you just brought in that first cycle. You'll lose half of them in the second cycle and then the other half in the third cycle on average, because at the point, although the enzyme can tell it apart right now, once we get to that step with the succinate, at this point, this is a symmetrical molecule. So it doesn't look like it right now, but if you were to think of the COO being right here, now remember when we see a COO, that's just a carboxylate group. So we have this molecule that's symmetrical, and at this point, the enzymes can't tell apart which carbons came from where. And so this is why in the next cycle, you'd lose the equivalent of half of it, and then in the next cycle, you'd lose the equivalent of the other half of it in terms of the carbons. So it'll take 3 cycles on average to get rid of the 2 carbons that you brought in from the acetyl-CoA, even though you lose 2 carbons each cycle. The first cycle through, though, you're never going to lose carbons from the acetyl-CoA you just brought in because the enzymes are able to tell it apart. But then once you get to this symmetrical stage, now your enzymes can't tell apart what came from where. And so things kind of get scrambled. Another couple of notes. So there are a couple of steps where you sometimes will see intermediates. So especially in the cis-aconitase. The aconitase step overall is an isomerization reaction, but it's going to be happening by first dehydrating and then rehydrating the molecule, generating a double bond that then you can add water across and get the OH group moved to the other position, setting yourselves up so that when you were to oxidize this, we would have this carboxylic acid be alpha-beta-2, that's ketone. When we lose the water here to form the double bond, this is not a redox reaction. Because we're losing the water, we're not just losing the oxygen. So we're losing an oxygen, but we're also losing a hydrogen. They kind of like cancel each other out. This isn't a redox reaction. We're just removing water. When we remove that water, we form a double bond. And then we can add water back, but add it back in a way that's going to put the OH group on the other position, in position, so that this, if we oxidize it to form a carbonyl, that carbonyl is going to be alpha-beta-2 the carboxylic acid, and that's going to make it favorable to decarboxylate. Why is it going to be favorable to decarboxylate? Well, here it gets back to that keto tautomerization that we saw in the case of our PEP carboxykinase, that we saw in the case of glycolysis with that big bang at the end of the glycolysis with our pyruvate kinase step. How by trapping the pyruvate, by trapping this in this kind of like enol form, where we have this, what would be an alcohol next to an alkene, that was unfavorable because oxygen would rather have those electrons than the carbon. If you release that phosphate group, now you can have tautomerization. The electrons can flow back to being bound to the oxygen to be your keto form. That was going to be highly energetically favorable. We have a similar situation here. We were to oxidize this, which we do with our isocitrate dehydrogenase. That'll give us this intermediate oxaloacetate. This is going to decarboxylate because beta decarboxylation is so favorable, because what happens is that it's going to leave you with this enol that is so unfavorable that's going to drive the formation of the keto, which is then going to make this whole process very favorable. If you think about the electrons in this bond flowing here, then pushing these electrons here, you would end up with an enolate. And then if you were to reverse things and let those electrons flow back, now you get back to the keto form, and this flowing back to the keto form is going to be what's going to be energetically favorable in driving this process. And so this is going to be a highly energetically favorable reaction, this isocitrate dehydrogenase, and we're going to run to regulate it tightly. And note that here we're going to have intermediates too. We're going to have like the oxaloacetate intermediate, but because the decarboxylation of a beta keto acid is so favorable, you're not going to actually see this intermediate. So it's going to be very, very, very transient, as opposed to the cis-aconitate, which is going to be more stable. Depending on where you look, it either can leave the active site transiently or it doesn't leave the active site, but it's going to be a little more stable than our oxaloacetate, where we have this highly energetically unhappy beta keto acid that would be very, very happy to get rid of that CO2 and therefore generate a temporary enol that then gets pushed to the keto and then makes it really, really hard to go back from. Each turn of the cycle, we're getting 3 NADH, 1 FADH2, and 1 GTP. If we were doing this from glucose, we'd be doing this cycle 2 times because we'd have 2 acetyl-CoAs because we had 2 pyruvates. We can keep doing this cycle as long as we keep feeding in acetyl-CoA. And we don't take these other things away. And because acetyl-CoA can't go through these mitochondrial membranes, we're going to need to do this oxidative decarboxylation of our pyruvate inside of the mitochondrial matrix. So we do glycolysis in the cytoplasm, then we take that into the matrix of our mitochondria in order to carry out this oxidative decarboxylation. Then we have the acetyl-CoA right in the right place for doing the citric acid cycle, because that's going to be taking place in our mitochondria. Later, we'll see how we also need to be thinking strategically about what we can take in and out of the mitochondria, because if we want to use products from the citric acid cycle, and we need to use those products in the cytoplasm, we're going to be having to take them out of the mitochondrial matrix And so if we can't take that molecule directly out of the mitochondria, we're going to have to take something else out of the mitochondria that then, once we're in the cytoplasm, we can revert back to the thing that we really wanted. We can keep doing this cycle as long as we keep feeding in acetyl-CoA. And we don't take these other things away. But the real fun of the citric acid cycle comes when you do take things out. When you take out malate to make your glucose, when you take out citrate to make your fatty acids, when you take out alpha-ketoglutarate to make glutamate, when you do all these things, you're able to use these parts from the tricarboxylic acid cycle to do all sorts of things. But if we want to then keep the cycle going, we're going to have to feed back into the cycle. And so then we'll take things like take glutamate and make alpha-ketoglutarate. We can break down our glucose and give us pyruvate that, yes, we can make acetyl-CoA, or we could also make oxaloacetate using pyruvate carboxylase. These are all going to be examples of anaplerotic reactions, and we'll talk more about these reactions. But the important thing to know is that you can go in and out of this pathway, and it's not just about that acetyl-CoA that you got from your pyruvate, that you got from your glucose. It's about all these different components that you got from all of these different places and that you can use for all of these different things. That's really why it is a central hub, that it is so, so, so awesome, so cool. I just want you to understand and appreciate how really, really freaking awesome this tricarboxylic acid cycle is. And it's so awesome that we really need to regulate it. And so you'll see that we have a lot of these steps are going to be highly regulated. And these key steps that we see regulated, these are going to be those steps that are going to have that really large negative delta G that are going to be hard to reverse. This is going to be our pyruvate dehydrogenase step, our citrate synthase step, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. It's not that these other steps aren't controlled by allosteric modulators at all. And so we know that malodehydrogenase, for example, is controlled by various allosteric regulators, so small molecules that bind and alter its activity. But these are kind of like the big ones. And you'll see that things like NADH, we talked about how it inhibits pyruvate dehydrogenase, but it's also going to inhibit your alpha-ketoglutarate dehydrogenase and your citrate synthase. Because, well, this process is going to be generating a lot of NADH, and we're going to need a lot of NAD+. If we have a lot of NADH and not a lot of NAD+, well, then we're not going to want to keep going down this pathway. Maybe that's indicating that we've got a buildup in our electron transport chain or something. Then we could say turn to fermentation in order to regenerate NAD to at least keep glycolysis going. And once we get rid of that backlog of NADH, well, then maybe this can continue again. This is going to be stimulated by ADP. And so if you need energy, you want to do this. So ADP and AMP are going to help stimulate this cycle. NADH is going to inhibit it. ATP is going to inhibit it. We'll look more at this inhibition, but just kind of start thinking logically about when might you want to do this, when might you not want to do this. It's a little complicated when you think about when you might or might not want to do the citric acid cycle because it does have anabolic and catabolic uses. But the nice thing about the cycle too is that this side of the cycle is kind of easier to go back and forth. And so by controlling the concentrations of these compounds, we're able to help control the cycle. A key example of this is going to be our malic dehydrogenase step. So remember that in vitro, we study it in the oxaloacetate to malic direction because it is more energetically favorable in that direction in vitro, but under standard conditions. But in the citric acid cycle, it's able to be energetically favorable. It's able to have a negative delta G because you're keeping the oxaloacetate concentration low. You can keep it low by bringing in more acetyl-CoA and reacting it with it. But if you don't have acetyl-CoA, you're not going to be able to do this. If you have too much acetyl-CoA, well, then you're going to want to stop making acetyl-CoA, and so that's when you would inhibit pyruvate dehydrogenase. But you're never going to be in a situation where you have a lot of oxaloacetate. It's all in comparison to the amount of acetyl-CoA. But the amount of oxaloacetate is purposely kept really low. And it's kept really low by keeping it from getting in or out of the mitochondrial matrix. Because of this, that's why we had to go through our complicated gymnastics of taking the malate out of the mitochondrial matrix if we wanted to convert it to phosphoryl pyruvate, if we wanted to do gluconeogenesis. Similarly, we're going to take citrate out of the mitochondria instead of taking acetyl-CoA out of the mitochondria if we want to make fatty acids, because we can't take acetyl-CoA out of the mitochondria, as we talked about in this video. This is all setting the scene for talking in more detail about the regulation of this pathway and some of these anaplerotic reactions, as well as how we can use components of this pathway for various purposes. So, tricarboxylic acid cycle, citric acid cycle, Krebs cycle, whatever you want to call it, as long as you love it, all's good.